Hi there, my name is Shaun Mahony from Penn State, and today I want to talk about a new multi-mapping read allocation framework called Allo that we've recently developed in the lab. So multi-mapped reads are reads that align to multiple places on the genome identically. These are a relatively large component of short read-based regulatory genomics assays, such as ChIP-seq, ATAC-seq, cut and run, etc. Up to about 30% of reads can be MMRs in those types of experiments. Current bioinformatics pipelines will either completely discard those reads or randomly assign them to one of their alternate mapping locations. And this, either ignoring this data or inaccurately assigning them, can have quite a big impact on our understanding of the role of repeats in gene regulation. For example, we know that repeats, transposable elements, and other repeats can contain transcription factor binding sites, even by looking, as these studies do, only on uniquely mapped reads. So we know that there's a high potential for more gene regulatory sites to be discovered within these regions. So the question is, can we correctly figure out where these MMRs should map to? One approach that was proposed quite a while ago by Sundos Kellis and others is that you could probabilistically map these reads. So for example, these MMRs are more likely to be generated from regions that are peaks within these experiments that contain other nearby reads. So for example, we could possibly probabilistically map these MMRs based on the density of surrounding uniquely mapped reads. The locations with more uniquely mapped reads are more likely to be the correct location. So Allo implements this approach, but it also implements another module using a neural network that takes advantage of another feature of this data. So this neural network tries to classify whether a location displays the read distribution shape that would go along with a peak location. So based on probabilistic read mapping and this convolutional neural network to predict the peak shape or the peak-like shape of an alternate mapping location, together these scores combine to give a weighting for each alternate mapping location for the MMRs. So eventually we use this weight to just choose a single mapping location for every MMR. So how do we know if this approach is accurately assigning these MMRs? So what we can do is take ChIP-seq data or ATAC-seq data or DNA-seq data that have relatively long reads, so say on the order of 100 base pairs long, and we can artificially shorten that data down. So if you think about the full length data, there's going to be some set of uniquely mapped reads at the full length that after we shorten those reads down will now map to multiple locations on the genome. But importantly, we now have a ground truth mapping location for those MMRs. So we can use those to assess whether MMR allocation is working or not. So for example, if we compare Allo's accuracy versus just completely randomly assigning those MMRs, we can see that Allo does quite a bit better. And Allo's combination of probabilistic read mapping and the neural network does better than just using probabilistic read mapping alone. So we've applied this approach to several regulatory genomics data sets at this point. So this is one example, applying it to CTCF ChIP-seq data from ENCODE K562 experiments. You can see that Allo finds or enables the finding of thousands of additional peaks. These are regions that are basically devoid of signal using UMRs, uniquely mapped reads alone. But you can see that there's signal after Allo allocates the MMRs. These regions contain, many of them contain the expected motif for CTCF, and they're often located near TAD boundaries. So we think that they're real CTCF binding events. Allo not only discovers these sort of new, or helps to discover these new binding sites that were not detectable with UMRs, it'll also help improve the resolution or the sequencing coverage of peaks that were found using UMRs. So thereby improving the resolution of the ChIP-seq experiments overall. So that's one example with CTCF, but we've applied it to several hundred ChIP-seq experiments from K562 cells, reanalyzing a lot of ENCODE ChIP-seq experiments. The median improvement in the number of peaks, or the median increased number of peaks that we discover in these data sets is only about 5%, which doesn't sound like a lot, but overall, we have discovered about 385,000 new transcription factor binding sites in K562 cells by incorporating allo into these experiments. So these peaks are particularly enriched at relatively young and relatively long transposable elements, for example, SVAs and ERVK elements. And the other annotation that we've noticed that allo peaks are particularly enriched nearby are nearby duplicated genes, so recent gene duplicates. These are obviously locations where evolution has recently duplicated genes, so they're potentially important and potentially important for disease states. But we think that current pipelines are also underestimating the types of regulatory signals that are detectable around those gene duplicates, and allo allows discovery of those signals. So in summary, allo, we believe, is quite accurate, an accurate approach for allocating these multi-mapped reads. It's quite easy to incorporate into pipelines. It's a BAM input and a BAM output, so you can insert it in any pipeline. We've tried to make it as easily installable and usable as possible. So the code you can get by just scanning this QR code here or going to this GitHub. It's also installable on Conda and pip. There's a manuscript appearing soon in Genome Research. So this work is primarily that of Lexi Morrissey's in my lab with assistance from an undergrad, Jeff Shee. And this work is supported by NSF and NIH awards. Okay, thank you.